Hi. Of course, it's the first day. This room is full since I've been here. Yeah, so as Ostaizka said, my name is Elsa. I'm from the US, but I study in Iceland, and I've been here since September. But I am pretty quiet, so if this is my first time seeing you, hi. I'll be honest with you, I'm terrified to give this presentation because I have no real background in the subject matter before I got here. So I'm studying marine and coastal resource management, actually, and this is my university. It's very small. There are 40 of us. It's a repurposed fish processing plant, and it's up here in Eastfjords, Iceland, and it's really lovely, but it's very different than this environment. And so, yeah, but let's talk about dogs now. Okay, so yeah, this is the official working title of my master's project, and it's investigating the role of pack management on the intestinal microbiota of Greenland sled dogs using a One Health approach. Okay, so the Greenland sled dog is a breed of working dog that's unique to the Greenlandic Arctic, and it's been used for centuries and generations of people. But what's most important for my thesis is that they're managed as a natural resource or a coastal resource instead of how you may imagine managing groups of dogs. And so what I mean by that is that dogs are used to bring hunters to the open water or the sea ice where they can hunt whale and seal as well as fish. So that's the dog's primary function. And then to add an extra coastal element, they're also fed a portion of that game that the hunters bring back. So they're fed these coastal resources, and that's also a reflection of the food that these hunters are bringing to their communities, so a portion of the community's diet as well. The dogs are also housed outside, just on long chains, where they can drink water from puddles and eat melting snow and ice, so they have a really high degree of environmental exposure. And in the summer, when they're not used for pulling sleds and things, they'll often try to supplement their diet because they're not fed as frequently by scavenging around the environment, whether that be like an animal carcass or human garbage as well. So these two pictures kind of show everything. We see the dogs go into the sea ice so that the hunter can hunt, and then we see the hunter feeding the dogs seal from the most recent hunt. So I wanted to understand what the role of this management strategy has on the dog's health themselves. And to investigate that, we looked at fecal samples from two populations of sled dogs in East Greenland. Our first half of samples came from Takatuormit, where the pack is managed as I just described in this classical management strategy. They're owned by civilians. Their diet is primarily marine mammals, and they have that high degree of environmental exposure. Notably here, they also don't receive any form of formal veterinary care, so these dogs have never received like a vaccine sequence irregularly, antibiotic treatment, deworming, any types of things that you would bring your dog to the vet to have. And then the second half of our samples come from Danneborg, which is the site of the Danish military base Sirius. So these dogs are owned and managed by the Danish army, and they're fed a high protein blend of commercial dog food. They have their own veterinarian that can treat them for anything they may need. And they have more limited exposure to the environment. So their enclosures outside are fenced in, and they're given clean drinking water. And the exposures have a very controlled cleaning regimen as well. So to skip over almost everything, I followed the EHI pipeline all the way from the sample collection to the lab work, the bioinformatics and the analysis. Nana did my lab work, so thank you so much. And there are so many people in this room who did 99% of my project. When I showed up in September, I only had the analysis to do, and that was a lot in and of itself. But this is what we found, this crazy tree of 555 mags, of which around 44% had no species level annotation, and that spanned 12 phylum and 139 genera. So that's our overview. But then when we start to look at the two groups individually and compare them, we can start to see some differences. So though we found no significant difference in the alpha diversity, we did find quite a notable difference in the beta diversity within our samples, indicating that compositionally these dogs have different microbiota. And if we look into that a little bit more, we see that 430 of our 555 mags are shared between both locations, and then there are 88 that are only found in the Atokatormit dogs and 37 that are only found in Danaborg. And of that 88, 25% of the mags only found in Atokatormit can be pathogenic, and they include some pretty important zoonotic bacteria like Clostridium novi and Enterococcus herae, which are associated with severe infections in both dogs and humans and have been shown to be zoonotic in the past. And in Danaborg, none of the mags only found there have zoonotic or pathogenic potential. This pattern of increased pathogenic potential in Atokatormit continues when we look at differential abundance of those 430 mags found in both locations. Yes, it works. Okay. So I've highlighted here two on the screen. We did probably 1,000 different differential abundance calculations, and they found a wide range of results with spanning from 58 genomes that are differentially expressed to 181, but they all had this same theme. And we decided to only use results that were found in multiple. And we see that, again, pathogenically important mags are found in greater abundance in Atokatormit. And I've highlighted on the screen E. coli, but then also Campylobacter upsaliensis, which again is connected to gastroenteritis in dogs and humans and can be pathogenic or zoonotic between the two. But if we move away from the taxonomic description and into the functional description, we used a DistilR to distill functional annotations from mags, and DistilR uses, bear with me here, a curated database of known functions that are important from the host perspective. I'll be honest, I have a very surface level understanding of this, so please talk to Anton if you have questions. But it aggregates these functions at three hierarchical levels, with the domain level being the most broad and the compound or element level being the most specific, and the functional level as kind of the intermediary between the two. If we look at the functional level, we found these 12 differentially expressed functional traits. And I've highlighted a few that are important for our talk today. We see elevated lipid degradation and polysaccharide degradation in the dogs in Atokatormit. And we see the same pattern of elevation in alcohol degradation and antibiotic degradation in Atokatormit as well. So this is where we can start to see some interesting trends that could be related to their diet and environmental exposure. So if you remember from the beginning, the dogs in Atokatormit are fed a diet of a lot of whale and seal, which are both high-fat animals, so we would expect to see this elevated lipid degradation. And our samples were also collected in August, so it's the summer period, the dogs are not in use, so they'll be trying to supplement their diet with anything they can, potentially kitchen scraps. That could be an explanation of the elevated polysaccharide degradation. Then the two in green may be more related to environmental exposure, either through persistence in the environment or interactions with human garbage that they're having. But when we go down one more level to the element level, so it's like the bottom of the triangle, we can see a bit more information here. So as I mentioned in the previous slide, we know that polysaccharide degradation is elevated in Atokatormit, but when we look more closely at which elements and compounds in particular are elevated, we see that pectin degradation is much higher in Atokatormit. And pectin is found really commonly in the peels of fruits and vegetables. So this again could be a reflection of the fact that they're scavenging through garbage. And if we look at a few more examples at that element level, we get a bit more information. So in the top left, we see glycerol degradation is elevated in Ithaca torment. Glycerol really commonly found in soaps and cosmetics and things like that, and they could be interacting with that through human waste and scavenging again. And then we can also see the 2,3-butanediol elevated in Ithaca torment, the degradation of it. And this compound comes from the fermentation of polysaccharides and is really commonly found in rotting fruits. So this is also a strong suggestion that they may be supplementing their diet with the kitchen scraps. I also just wanted to show that we can see which specific antibiotics may be increased in their environment or that they're interacting with. We have tetracycline and streptogrammin. And again, we know these dogs are not being treated with antibiotics, so it must be coming from another source. And whether it is persistence in the environment or interacting with human waste, we can say that even if it is garbage, that's likely making its way into the coastal environment, which would have implications on the whole coastal system. And so I think these pictures kind of sum up everything. So we see that the dogs in Ithaca torment are being fed whale, this is whale, and it's a reflection of them being fed those coastal resources. And then here we also have those same dogs with that high degree of environmental exposure. So you can see the untreated water that they're living in and drinking from, but then up the creek a little bit further, we see a pile of garbage. So they have that, the ability to scavenge, and any compounds that may be in the runoff from that are going to the dogs. So I've about a month left to write everything up, and I'm now going to be zooming out of the pretty heavy microbiome analysis to be discussing this using the One Health approach, which is a theoretical framework that combines wildlife health, environmental health, human and domestic animal health as one holistic system. And the idea is to show first the value of using these new molecular tools in management decisions, as well as to highlight the importance of including microbiome research in coastal studies. So yeah, I'll take any feedback you have. Thank you.